would really like to spend a couple of minutes here digging into the downstream considerations as well for mRNA. I think it's an interesting concept, right? Because we're still trying to figure out from a quality standpoint where the FDA, what we're going to need to demonstrate to the FDA in terms of quality. It will have to be a high quality product, but what does that mean, right? In terms of an mRNA therapeutic. And so we've just talked about some of the optimization, some of the considerations we have to think about on the upstream to make things from an older sibling standpoint, right, easier for the younger sibling or the sibling, the second twin to follow, right, in footsteps. So there's where I see some of the greatest process differentiations, and I'd be curious to hear your thoughts as well, is in the purification process. Is that accurate, first of all, to say, is that where you're seeing some of the greatest differentiation, right, in terms of process? And do you have a general sense as to why that might be the case? We can start with John here.
Yeah, it's a good question. I would say that I do think you see variation, only because if you look at, again, your upstream process, there may be perhaps differences, right, across the industry as far as the purity that you're achieving at that particular part of the process, right? So you have to remember whatever you have to remember what the intent of the purification process is, right? You need to clear your nucleotides, there's specific proteins. If your upstream process has chelating agents, okay, you need to demonstrate clearance, chaotropic reagents, et cetera. So I do think there's some variation in the upstream process that leads to subsequent, differences in the downstream process. Yes. I think fundamentally, you use standard affinity chromatography processes, looking at different resins that are designed to bind and elute the mRNA. I know everybody's looking to remove double-stranded RNA. I think that's a simple example, and there's resins that are specifically designed for that. But you have to look at the totality of your inputs and your impurities as you do your characterization to optimize your downstream process. So I think the variation of the upstream process is likely driving the creativity in some of the downstream purification steps.
I like that. The creativity in the downstream. I like that. That's a great way to put it. I'm going to steal that someday. Hari, do you have anything to add? I think John really nicely summarized the challenge at hand, right? In terms of just because we can make a lot of mRNA, right, from an upstream process, there are going to be inherent purification differences as well that require.
He did an amazing job. Absolutely agree with him in terms of the creativity of the upstream requires different creativity of the downstream. And so
there's a terrible poetry metaphor to be made in here. I'm like really excited.
Not much long suit. The beauty of our industry is that there are many ways to get to the final answer, and the tools and techniques that are available are quite diverse and multitude of them, and so that leads to creativity. There may be a future where we will end up converging like we have in the case of monoclonal antibodies, where protein A and followed by photography steps. I think in one of the early stages of mRNA, and so the creativity is in full force at the moment.
Yes. Well, some of it too, are we really just trying to figure out, right? I think one of the bigger questions that I also have is thinking about the purification technologies we're using, trying to figure out what might be the best ones to rely upon. Is that an accurate assessment, or are we really, is it going to all come down to that upstream, and you think that creativity will live long into the downstream processes?
Yeah, I think so. I mean, I think, as John mentioned early on during this session, upstream drives a lot of what the downstream ultimately ends up doing. And so if IVT is appropriately engineered, and as I mentioned, we're still in the early stages of fully understanding and engineering that step. Once that's complete, then you can imagine a fairly abbreviated downstream process, hopefully. And so the success of downstream is actually buried in the success of upstream in some ways.
Yeah.
And so, yeah, I think we have ways to go before we get to that.
I think one of the challenges, and I sort of intimated this earlier, is, you know, we're really trying to determine how pure is pure enough, right? As one of the bigger questions of this industry. And so I'd love to hear both of your takes on how you're approaching that question, right? Especially as we're sort of living in an area where regulatory wise, we don't have huge overarching guidances, right? It's relatively piecemeal. We hopefully are working closely with the agency to get the answers we need internally. But, you know, I'd like to start with John here. How do you, or how are you defining what the best purification strategy is for your mRNA products when we really are still trying to figure out how pure is pure enough? Do you have any thoughts or guidance to lead us to clarification on that?
Yeah, from my perspective, I think the mentality is we should always be on a journey of continuous improvement, right? So I think it starts there, right? If you ask what pure is pure enough, okay, keep driving to a higher level of purity. Just keep going. Know the journey this is a journey that doesn't end, right? But from a more pragmatic perspective, I think what's important from my perspective is that you characterize your impurities and you understand what they are, right? I've historically seen companies or scientists kind of stop short of really understanding, hey, we see some impurity peaks, we have problems separating those peaks, and we stop short of identification. You need to identify what your peaks are, and then based off of that, you need to do an assessment. I think in mRNA, it's very clear. There's, double-stranded RNA is one impurity that we certainly need to clear, and because of the potential impact from immunogenicity perspective.
Yes.
But when we look at tailless species as example, there's plenty of literature out there that suggests that would be tolerable. It's not, it does not have biorelevance and gets cleared very quickly, right? So I think it's really applying that good science up front, and then always remember that you're on a journey where you need to characterize your process, and then you should always be driving continuous improvement. So I don't have I have specific numbers. In short of sharing specifications, I think that would be the directional answer that I can provide.
Darn, I almost got you some specs, guys. Almost. Hari, what any additional thoughts on what John really nicely summarized here for us?
Yeah, absolutely. I guess maybe provide another perspective, which is the classic quality by design kind of perspective. To start out with your target product profile, then you get to your quality target product profile, which tells you what your product needs to be, and then that gets to your control strategy as to how you're going to get to that QTPP. And so it starts always with what product you're trying to make and what are the requirements of that particular product, right? And having a very clear view on that determines your purification targets. Ultimately, what you want to know is what are the impurities and what are the pharmacological impacts of those impurities, whether it's from a safety or an efficacy perspective. And defining that up front drives you to what purification process and what specifications you need to meet at the end of it.